topic, please like, share, and subscribe the channel. In the last session, we dealt with quantitative procedures of proteins. In today's session, we will deal with proteomics and enzyme. Proteomics is a study of the entire galaxy of proteins produced by a cell under different conditions. At a particular time, a gene is on in a particular cell, but it will be off in another cell. Expression of proteins during growth and development will be different from the resting cell. Proteins produced by gastrointestinal cell and a neuronal cell will be entirely different. Many proteins are getting post-translational modification, that too at different levels at the various organs. But genes are the same in all cells at all times. Therefore, genes or the study of genomics will give only a partial picture of what is going on in nature. Human body contains hundreds of different cells which express thousands of proteins at different times under different influence of different stimuli. The proteomics attempt to study all these multifaceted picture on in toto. Life is enzymes are biocatalysts. Life is possible due to the coordination of numerous biologic reactions inside the cells. Proteins can be hydrolyzed with hydrochloric acid by boiling for a very long time, but inside the body, with the help of enzymes, proteolysis takes place within a short time at body temperature. Enzymes catalysis is very rapid. This usually one molecule of an enzyme can act upon 1000 molecules of the substrate per minute. Lack of enzymes will block in metabolic pathways, causing inborn error of metabolism. Substance which upon which an enzyme acts is called the substrate. The enzyme will convert the substrate into the product or products. Characteristics of enzymes. Almost all enzymes are proteins. Enzymes follow the physical and chemical reactions of proteins. They are heat labile. They are water soluble. They can be precipitated by protein precipitating agents like ammonium sulfate or trichloroacetic acid. They contain 16% weight as nitrogen. Classification of enzymes. When early workers isolated certain enzymes, whimsical names were given. Some of these, such as pepsin, trypsin, chymotrypsin, etc., are still used. Later, it was agreed to call the name enzymes by adding the suffix "is" to the substrate. Thus, enzyme lactase acts on the substrate lactose and products glucose and galactose are formed. Enzymes that hydrolyze starch, that is amylose, are termed as amylases. Those that dehydrogenate the substrate are called dehydrogenases. These, no, these are known as the trivial names of the enzymes. IUBMB classification of enzymes. International Union of Biochemistry and Molecular Biology IUBMB in 1964, modified in 1972 and 1978, suggested the IUBMB system of nomenclature of enzymes. It is complex and cumbersome but unambiguous. As per this system, the enzyme starts with EC, that is enzyme class, followed by 4 digits. First digit represents the class, second digit represents the subclass, third digit represents subclass or subgroup, and fourth digit gives the number of particular enzyme in the list. The enzymes are grouped into following 6 major classes. Class 1 is oxidoreductases. This group of enzymes will catalyze oxidation of one substrate with simultaneous reduction in of another substrate or coenzyme. This may be represented as AS2 plus B gives A plus BH2. For example, alcohol plus NAD plus gives aldehyde plus NADH plus H plus. The enzyme is alcohol dehydrogenase and IUBMB name is alcohol NAD and oxidoreductase. Code number is EC1.1.1.1. Oxidoreductases may also oxidize substrates which by adding oxygen. Example oxidases, oxygenases and dehydrogenases. Class 2 will be the transferases. This class of enzymes transferred one group other than hydrogen from the substrate into to another substrate. This may be represented as AR plus B gives A plus BR. For example, hexose plus ATP gives hexose 6 phosphate plus ADP. The enzyme, the name of the enzyme is hexokinase and systematic name is ATP hexose 6 phosphate transferase. Class 3 is hydrolases. This class of enzymes can hydrolyze ester, ether, peptide or glycosidic bonds by adding water and then breaking the bond. Example, acetylcholine S plus H2O gives choline plus acetate. The enzyme is acetylcholine esterase, which is the trivial name, and acetylcholine hydrolase is systematic name. All enzymes are hydrolases. Digestive enzymes are hydrolases. Class 4 is lyases. These enzymes can remove groups from substrate or break bonds by mechanism of other than hydrolysis. For example, fructose 1,6 bisphosphate gives glyceraldehyde 3 phosphate plus dihydroxyacetone phosphate. The enzyme is aldolase. Class 5 is isomerases. These enzymes can produce optical, geometric, and positional isomers of substrates. Racemases, epimerases, cis-trans isomerases are example. Glyceraldehyde 3 phosphate gives dihydroxyacetone phosphate. Enzyme is triose phosphate isomerase. Class 6 is ligases. These enzymes link two substrates together, usually with the simultaneous hydrolysis of ATP. In Latin, ligar means to bind. For example, is acetyl CoA plus CO2 plus ATP gives mannose CoA plus ADP plus PI. Enzyme is acetyl CoA carboxylase. Coenzymes. Enzymes may be simple proteins or complex enzymes containing a non-protein part called the prosthetic group. The prosthetic group is called the coenzyme. It is heat stable. The protein part of the enzyme is then named the apoenzyme. It is heat labile. In this diagram, we can see that apoenzyme or the protein portion or the inactive part combines with coenzyme or cofactor or non-protein portion or prosthetic group to give holoenzyme or the whole enzyme. These two portions combined together is called the holoenzymes. Coenzymes may be divided into two groups. First, those taking part in the reactions catalyzed by oxidoreductases by donating or accepting hydrogen atoms or electrons, and those coenzymes taking part in the reactions transferring groups other than hydrogen. In the first group, the change occurring in the substrate is counterbalanced by the coenzymes. Therefore, such coenzymes may be considered as co-substrates or secondary substrates. The substrate lactates is oxidized and simultaneously the coenzyme co-substrate is reduced. If the reaction is reversed, the opposite effects will take place. Nicotinamide adenine dinucleotide. This coenzyme is synthesized from nicotinamide, a member of vitamin B complex. The structure of NAD plus is NAD ribose phosphate, phosphate ribose adenine. Warburg, who was awarded Nobel Prize in 1931, elucidated the structure of NAD plus. Reversible reactions of lactate to pyruvate is catalyzed by the enzyme lactate dehydrogenase, but the actual transfer of the hydrogen is taking place on the coenzyme NAD+. In this diagram, we can see that lactate is converted to pyruvate in the presence of enzyme lactate dehydrogenase and conversion of NAD plus to NADH plus. In this diagram, we can see that NAD plus is converted to NADH. In this case, two hydrogen atoms are removed from lactate, out of which one hydrogen and two electrons are accepted by the NAD plus to form NADH, and the remaining H plus is released into the surrounding medium. The hydrogen is accepted by the nicotinamide group. Here ends our topic. Please like, share and subscribe the channel. Thank you.